Okay, so benvingudas e benvinguts. Welcome, everybody. I'm Camille Stefan. I'm the head of the Biostatistics and Bioinformatics unit at IRB Barcelona. So I can officially welcome you from Catalonia, not from Sitges, but it is, I guess, close enough. We are very excited to have so many people registered. Hopefully, more people will join as we advance. And I've known this event since 5 or 6 years now, and I really like it. And basically because I think it creates a community of people that are working in biostatistics course or bioinformatics, but also users. And so it's very interesting to see both points of view and also to share topics as diverse as budget, managing, data managing, methodology. So I'm really excited. If I didn't help organize this, I would definitely join. We have super exciting talks today. And hopefully we're gonna be able to have interaction and a very dynamic workshop. So we're gonna have 40 minutes talks more or less, and then time for questions and answers. So about that, I want to point you to the tools we have in the platform for those of you that didn't use it before. So we have a discussion forum where you can interact with other participants. You can introduce yourself, which I think it's really interesting to know who we are talking to. And then we have the live questions and answers, where you can directly answer, ask questions to the speakers. You can also like these questions so that your question goes up in the interest rating. And also we may decide to run or to launch a poll where you can vote for the options we give you. And lastly, but very important, there is a link to the Slack channel from the European Bioinformatics Core Community Workshop. And hopefully we can start interacting now, but also continue doing so. So this is a permanent channel. So hopefully we'll have a lot of new connections and interactions over there as well as here today. So I don't wanna take any more time. So Dr. Sven Nansen will chair this first session. So Sven, the mic is yours. You're muted, Sven.
Break. We will start part 2 of the workshop, and I will hand over to Professor Chris Evelo to chair part 2.
Thank you very much. I'll do that. I will introduce Sarah Hunt very briefly. I think the previous talk is actually a great introduction for what we try to do in the European Joint Project on Rare Diseases, where we try to work with clinical experts on rare diseases, organize their data, where we have the same types of problems, essentially, like a lot of different types of omics data, and then trying to predict either the disease diagnosis or suggest treatments from that. And we have, of course, if we then do that analysis, we stumble on things like, okay, we see a lot of variants occurring in these patients. Can we somehow evaluate or even predict what the effects of these are, which is what Sarah will talk about. Sarah is, I'm really happy to have Sarah in that project and other great participants like the NextPlot group. She leads the variant effect predictive work at EBI. Sarah, please take the floor.
Chris. So as Chris said, I work on the Ensemble project at EBI, and we produce the Ensemble variant effect predictor. And today I'm going to give a quick guide to some of the resources available for variant interpretation in rare disease studies. And I'll talk about some of the considerations to take into account when using them. So as we know, rare diseases are individually quite rare, but there's a lot of them, so they're actually quite common. I think it's something like 1 in 7 people will suffer one at some time in their lives. So there's now many research groups focused on specific clinical presentations, but there's also national initiatives such as Genomics England, employing bulk sequencing and genome interpretation. So the rare occurrence of each individual syndrome means that data sharing is very important to understand the syndromes. And so standards are essential for communication and comparison of results. So here's a simplified process of how you go about analysing data. So you start with your variants, either from exome or genome sequencing in a BCF. You'll look at the gene annotation to predict molecular consequence. You'll look at allele frequency resources to identify and probably exclude common variants. Then you'll look at other resources for phenotype and citation data, so you're aware of whatever else is discovered about a variant. And there's a wealth of prediction packages to run to try and evaluate the pathogenicity of your variant. So you've then got a huge pile of information, you need to filter it, and you get to set a smaller set of variants and potential candidate genes. And then you can move on to look at gene function. So the Ensembl variant effect predictor started off as a very simple wrapper around the Ensembl API to map variants to transcripts and predict molecular consequence. As new data sets and algorithms have become available, functionality has increased, and VEP's now quite an extensive and sophisticated tool. So this cartoon here shows a transcript and some of the consequences we predict. So if you see they're based on position, such as the 5' UTR variant, splice donor variant, and also the actual change, so missense variant, which changes the amino acid. And VEP is built on the Ensembl databases, so I'll quickly say something about how we build them. Our human databases are built with a backbone from DBSNP with RefSNPs. We apply quality control and standardization to this, and then we import richer sources, such as positions of variants in HGMD, ClinVar association information, rare disease gene associations from OrphaNet. Then we integrate frequency information from resources such as TopMed, NOMAD, 1000 Genomes, and citation data from sources such as Europe PMC. We then analyze these data with respect to other Ensembl resources such as the GenCode transcripts, the regulatory data, and comparative analysis resources we have. Of course, we use ontologies to try and standardize descriptions wherever possible. And this information then is released via VEP, our website, Biomart, and our APIs. So to return to VEP, it's optimized to annotate variants in VCF format when they're ordered by genomic location. It can also deal with a number of identifiers from popular resources, but these have to be done through online methods. We can also accept HGVS at genomic, transcript, or protein level, though of course protein level HGVS can be tricky as it might result in ambiguous results. So as people working in core bioinformatics, I assume you're more interested in command line tools than web interfaces, but I'll briefly mention our web interfaces as they give an idea of the kind of information available, and they're quite handy for testing the results of one variant, say. So we have a web tool that takes a file of maximum about 50 meg, and this is about the size of an exome. Jobs here are managed on a scheduling system on a HPC farm. It's simple to use, but it's not very configurable. So this is an illustration of the kind of results we can make available. So there's summary information of what's been processed. There's a panel to filter your results. And then there's a table below the main part of the results is a list of consequences and other data per variant allele and genomic feature. So I don't know if you can see my pointer, but we've got links to transcript information, existing variants, and then if there are structures in PDBE, we can also show an interactive widget showing where the variant is on the structure. So we also have a REST interface that takes HGVS variant IDs and a position allele-based input. This is good for small amounts of variants. And there's a number of different options here, different tools and different data sets that can be retrieved. The most customizable and efficient way to use VEP is the command line interface. So here you've got total data privacy. Nothing needs to go out over the wire because we produce reference data sets that can be downloaded in a compact format that's quick to use. We produce Docker images and VM so that it's easy to install. There's also a Bioconda recipe. This has been made available by a third party. As I said, standards are very important. So back in 2015, we agreed a basic VCF output format with the authors of SNP-F and Anavar. It's been adopted by UCSC as well since. And we're now participating in global alliance efforts to create new models to describe sequence variants and their annotations. It's possibly early days yet, but hopefully next year we'll have something ready to use. So I said command line VEP is very extensible. One way we do this is plugins. So these are discrete modules that add specific functionality. So they're optionally added at the command line, and they can be written really quickly when a new tool or data set's available. So the common kind of thing these would do is run an additional algorithm, look up pre-computed values from a file, or even use the Ensembl API to pull out more information. So these are very handy. We incorporate public data where we can, but sometimes licenses don't allow that. So we can write the parsers as a plugin module, and then the analyst can pick up the data, agree to the license themselves, and it's very easy to use. So to go back to the process, first step, I'll start with gene annotation. So transcript choice has a huge impact on gene annotation. There are two main sets of annotation for human. There's the Ensembl GenCode set and the NCBI RefSeq set. And of course, because of tissue-specific expression, there are many different splice isoforms available for most genes. So choice of transcript set has a big impact on annotation. People sometimes don't want to wade through 100 transcripts to look for their variant impact information, but it's really better to consider the whole set. Both Ensembl and RefSeq supply default transcripts, but if you only use this in your analysis, you will miss information. Transcript quality information can be used to filter. So there's a number of options here. APRIS identifies principal isoforms for different vertebrate species using protein structure information, functionally important residues, and information from cross-species alignments. There's also the transcript support level, and this summarizes the amount of information available for a transcript. So having said you shouldn't only use the default transcript, I'll double back to say it can be very useful for reporting. And we can run into problems when different groups use different default transcripts. So Nomad has been using the Ensembl default, and ClinVar often uses the RefSeq default. So one of the things we've been doing recently is working with RefSeq to try and come up with a shared default. Our usual default pipelines are quite similar. So it's an automated pipeline, and there's a manual review step afterwards. And we come up with different results because though we use mostly the same information, we give different weightings to them. So there's now a team of curators on each side discussing what the ideal transcript would be. And you can imagine this was quite easy for the first, I don't know, maybe 50% matched already, but the rest, it's going to be trickier. So we're calling this data set the main set, and this almost is matched annotation from NCBI and EBI. The attributes here are, it will match the 38 reference completely. It will be identical in the UTRs and the CDS between both sets. And there's two tiers planned here. So the main select will be one transcript per gene, and this will be representative of the biology at the locus. So it'd be well supported, expressed, well conserved. So at the moment, 80.4% of human genes have a main select transcript in Ensembl. When we get to our autumn release, this will go up to 83.4%. So there'll be another set as well as this default set, and these are main plus. And these are alternate transcripts that capture some key aspects of gene structure or biology at the locus. These are usually requested by a clinical expert. So quite often this will be a fetal version and an adult expressed version. These will be going live in our autumn release, and there'll be 41 transcripts there. So we don't expect a huge number here. But we're hoping this becomes adopted as a clinical reporting standard. We're encouraging people to use these. So we take our transcript sequences and we translate them, and we run Interpro scan on them. So Interpro pulls together a variety of different protein, domain, and family databases. And with this information, you can see when your variant falls in a particular domain or family. So to summarize, the cache data we provide for download with each VEP release contains the Ensembl gen code and the RefSeq transcript annotation, so you can analyze against either or both. Transcript quality information, including the main status, APRIS, and transcript support levels. And also mappings to protein databases, so we map to UniProt and PDBE, and the Interpro features as I've described. So currently the UniProt mapping is to the accession. So it's from our protein accession to the UniProt protein accession. In our autumn release, we'll start mapping to precise isoforms, which should be more useful. And here you see in this output, we've got the transcripts listed. Where there's a main, we're listing the RefSeq identifier as well. We have the transcript support information, and we've got links out to SwissProt. So I should comment on the consequence terms we use. So as I've been saying, standards are very important. So we use the sequence ontology to assign consequences. And we've been working on developing this for some years with the sequence ontology team, and it's been adopted widely by the major databases and tool providers. Where possible, we always use the most precise term. So if a variant can be classified as a missense variant, if it changes the amino acids, then we wouldn't also class it as an in-frame variant or a coding sequence variant. Though obviously query tools allow you to pull out child terms when you specify a parent. I should also say that regulatory data is in the Ensembl cache. The regulatory build takes information from all the major consortia, and it produces a set of features. So there'll be regulatory regions, including promoters and enhancers, and then transcript factor binding sites. And we can report when a variant falls in these. So we now have our predicted molecular consequences. We can move on to look at variant information. So it's common to filter variants discovered in a case against population reference sets, as variants common in a healthy population are unlikely to be causative. So the ACMG uses 5% allele frequency as strong evidence that that allele is not pathogenic. In general, you want to pick the frequency that's less than the disease prevalence in the population. Of course, this only works if it's a single causal variant, and you need to consider variants acting together might be more common, but more rare in combination. So there's a number of good panels available now. The 1,000 genomes set was the first, and that has a good range of different ethnicities, and they're all healthy individuals so far as we know. Studies like TopMed came along later. So these people all have heart, lung, blood, and sleep disorders. Nomad, of course, changed everything with particularly deep data. So for the exomes now, it's over 125,000 samples are available. So that's pretty impressive. We're getting down to quite low frequencies. A new recent project is the Alpha project, and this takes data from dbGaP and extracts summaries. So at the moment, I think when I thought there are 100,000 samples worth of frequency data available, but this is meant to be rising to about 1 million over the next 2 years, which will be quite exciting. It's also interesting that we can finally look at dbGaP data after worries in the past with the Homer paper about re-identification. So when using frequency data, you need to be aware of the presence of cases in these sets, such as TopMed. I need to be aware of overlapping sample content, so TopMed is part of nomad genomes. And also you need to be aware of ambiguous locations for insertions and deletions that can confound mapping. So this shows something about how we would do some allele matching to compare input variants to a frequency resource. So I don't think anyone would now produce a VCF line quite so hideous as this, but it illustrates the approach. What we do is take each allele in turn, strip it and the reference to a minimum representation, and then look in frequency resources to find a match. So we now have checked against frequency resources, and we know what the common variants are and which have not been seen. We can move on to look at phenotype and citation data. So phenotype data can be available by gene or variant. Of course, you'd ideally want it by variant, because just because the variant falls in a gene with a specific phenotype doesn't mean that that variant would cause it. Data is highly heterogeneous, and some sources also contain other sources. So you can't assume that presence in two databases improves the strength of evidence. For instance, everyone imports OMIM. So we have a phenotype plugin to pull out information from the ensemble databases. Also here, I should mention LOVD, which is a network of gene-centric expert-created databases and good for pulling out variant-specific phenotypes. When dealing with variant-specific phenotypes, of course, you have to look at the alleles, because these days we're getting more and more multi-allelic variants. And as in this case, one variant with 3 different alleles. One is pathogenic for various cancers, and the others, there's been a study, but they're of uncertain significance. So one you would take seriously, and the others you wouldn't so much. So a lot of phenotype data is extracted from the literature, but of course, not all the literature has been curated like this. So citations can provide an additional source of otherwise unknown phenotype associations. So baked into the VEP caches, we have results mined from literature by RefSNP ID, and this is done by Europe PMC and UCSC. We also have citations that are submitted with phenotype associations, for example, to ClinVar. So you have to be careful using this kind of data, as typos are scarily common. So we apply some basic QC. For instance, we wouldn't import a citation for an RSID where the paper was published before DBSNP existed. We also tried to do some basic QC on the species to check it doesn't look like it's a paper talking about rice, which is unlikely to be an association to human. So there are other resources for citation data. So DisGenet employs curators to look at phenotype associations, and we have a VEP plugin for that. And Mastermind is a newer resource which pulls out HGBS style variant descriptions from the literature and has quite a huge set. So to summarize, variant data in the VEP cache includes known variant locations, frequency information, citations, and clinical significance assertions from ClinVar, which are reported by allele. So we realized that might not be all the information you want to use. So the VEP API can extract data from standard reference files to include in the annotation. So this would be transcript information in GFF, variant frequencies or other info from VCF, and genomic features in BED. So example use cases might be genome-wide data sets from nomad or TopMed or UK10K, because we only supply the exome sets in the caches, or maybe private data sets that you have. So we now have association information. The next thing to look at is predictions. So when dealing with prediction packages, the important thing is to remember they are predictions. So two good measures of whether a change will be tolerated are conservation across species and constraint within. Because if a region is the same across many species, then it's highly likely to be important. And if you're seeing less variance in a region, in a gene than average, then again, it's highly likely to be intolerable to change. So there's a number of measures of conservation. We have GERP as one of the ones we use in Ensemble, but PhyloP and FastCons can be added through custom annotation. Constraint scores by gene have been coming out of the EXAC and NOMAD projects. So the PLI score, the probability of being loss of function intolerant, is very popular. And there's also the missense badness polyphen constraint score, which is looking quite good. So there are now very many missense variant impact predictors. So performance depends on the type of data available, so it can vary by gene. Many of them use protein homology information, and for these, the predictions are heavily based on the alignment, so can change as the databases change. Many use the same training sets as well. For instance, HGMD is very popular. And many now incorporate other callers. So when you're seeing agreement between a number of pathogenicity predictors, it may be due to noise or the fact they're very similar. And it doesn't always increase the evidence. So one nice tool is the DB-NSFP. So this pre-calculates scores for many different predictors on all possible missense variants on a specific transcript set. So you can pull out whichever suits your requirements. So recently, a number of ensemble methods have been produced, and combining multiple predictions can provide a more robust scoring. So CAD is one of the most popular ones. This uses over 60 different features, including conservation, functional information, expression information, and protein level scores. And this is genome-wide, which is particularly useful. Revel is one of the newer popular ones. This is missense only, but I hear it works particularly well on in rare disease studies. It incorporates a wide number of different predictors. So of course, not all causative variants will be missense variants, and it's important to look at disruption to splicing. So one of the popular tools long-term here has been MaxEntScan, which uses a maximum entropy model to evaluate the probabilities that a sequence is a real splice site. There's another nice collection tool here, DB-SC-SNV, that pre-computes scores for 7 different predictors and produces a learning score based on that. And a lot of people are telling me they like Splice AI, which is a neural net tool, and this predicts splice sites deep into the intron and exon, which is an advantage. So we now have a VCF annotated with lots of information. We need to do some filtering. So how you filter your annotated variants depends on what you know about your syndrome and your hypothesis. So we provide a filtering script with VEP, and this allows you to build and apply custom filtering criteria. For instance, you could filter by ontology terms and pull out all the child terms of coding sequence variant. You could filter by low frequency or not found in a specific population. And you could also filter for a certain CAD score. If you're particularly interested in loss of function variants, then Conrad from the NOMAD team produced the Lofty plugin. So this takes stop gained, frame shift, and splice disrupting variants. It applies various different criteria to reduce them down to a set of probably very interesting variants. You may know more about the syndromes you're interested in. You may have gene panels. So one of the things we're involved with as part of the Transforming Genomic Medicine initiative is we've built a tool to create gene panel information. We've got 4 panels available publicly at the moment. And for these, we have built the G2P plugin. So for this, we take the proband VCF of genotypes and we calculate the molecular consequences as usual. We then use the gene panel information and find the mutational consequence we expect, like is the syndrome always caused by missense variants? So we can filter on that. We filter against population frequency sets based on whether we expect 1 copy of the gene or 2 copies of the gene to have to be broken to cause the syndrome. And then we filter the variants to find any that meet these criteria. Once we have the variants, we can look at transcripts that have the appropriate number of variants, and that gives us a list of probable genes. We can also, of course, include a variant inclusion list for those cases I mentioned where you have multiple variants acting together, so they may be high frequency but still of interest. So as I said, we designed this for our G2P curation tool, but it takes a simple format which other panel information can use. And we've tuned it to be very sensitive but less precise because we don't want to lose any information. So of course, short variants aren't all we have to deal with. Structural variants can be important, though they're somewhat harder to handle. So BEP annotates CMVs, deletions, insertions, and duplications in VCF format. So either the end or SVLen is required. And if we have that, we can report the proportion of coverage of any overlapping feature. For deletions, we can check for transcript ablations and truncations. For dupes, we can check for amplifications and elongations. And for insertions, we can check for elongations. So here's an example output from the web tool again. The structural variant in red completely overlaps the transcript. And so we get a transcript ablation consequence. So another one here, you can see from the list of exons and introns covered that both exons and the one intron is covered. So this is a duplication, so we get the transcript amplification status. So I said before, population frequency data is very useful. We now have population frequency data for structural variants, 2 prime sets of 1000 genome set on the NOMAD set. We have a plugin to incorporate this information into VEP analysis. It returns percentage overlap name and allele frequency. It's more complex with the structural variant, so you can tune how the overlap works. A default would be an 80% match returned. And we'd also match on sequence ontology class, of course. So our support for structural variants is quite basic at the moment, but the area is becoming more popular. So it's something we're planning on working more on in future. Something else we're working on a lot at the moment is the European Joint Programme for Rare Disease. So the aim here is to create a research and innovation pipeline from bench to bedside, ensuring rapid translation of research into clinical applications and uptake in healthcare and benefit for patients. So there's 35 different countries participating. And what we're trying to do in our part of it, it's a huge project, is improve integration and efficiency between different tools and processes. So one of the tools, so obviously VEP is one of the tools. Another one is NextProt. So this integrates knowledge on human proteins. It takes information from a variety of resources, including UniProt, interactions from Intact, the Human Protein Atlas, and a wide range of variant-specific annotations as well. It has browser views, so you can see if a variant lines up with a domain. And it provides access via APIs to build quite complex queries. So there's a Sparkle service, and this enables federation with other resources. So this is how Nextprot has been joined to DisGenet and Wikipathways, and it's also how we've set up a VEP plugin. So at this point, I'll pass over to Chris to talk about another EJP resource, Wikipathways.
Thank you, Sarah. Like I said at the very start, we want to evaluate data in rare diseases with respect to this kind of variants, and of course that means that we want to see again how they actually act together. And this is actually one of these biological pathways, like we described it before in the previous session, that was created for one of the rare diseases. This is Rett syndrome, which is known, I hope you can see my cursor, which is known to be focused about one gene, where most patients actually have a problem in that gene, but we know that gene plays a major role in all kinds of regulatory processes. It's actually an epigenetic gene. So you can build this pathway that connects all that stuff, and we have that on Wikipathways, which is just one of the, well, I wouldn't say many, but one of several pathway resources currently available, where the main difference with the others is that this one is not only open source, but also open data, in the sense you can even directly contribute to it. It's really a wiki. Can I have the next slide? Oh, the next slide. Yeah, right. So the important thing is that we actually have that information available for computational access. So there is an API, there is a Sparkle endpoint, and we actually, which means you can integrate it in resources, even like Ensemble, if you would want to. It's integrated in quite a few already. It's like you can look up the pathways that have information from that resource included in the pathway, and we provide you actually with the source libraries to then really work with these. Plus, you can actually download the content in different ways, which might mean that a lot of you have actually already worked with it without knowing that they did. Now, the real question is, of course, can you really use that in combination with things like Variant Active Predictor? And there I don't have anything to show you yet. Just the answer is yes, in principle, given that we have a plug-in mechanism where you could easily connect more information from another resource to genes in a pathway. What I would want to show to you on the next slide is what we did with this basic idea so far. Like, if we really have this idea that known variants relative to a known disease actually work in concert, then that should somehow show up if we start to connect that to pathways. Right? And can we then see that multiple variants really occur in one pathways? And can we actually start to use that to show more information about mechanisms and such? Can I have the next slide? Right, so this is work from Elisa Cirillo, who herself is actually a dancer, but she danced around in this whole spectrum of genetic variants. And what I will show you on the next slide is how she did that for a disease where we actually know lots about related studies, so many that people actually start to doubt how real that really is. Right? If you go into GWAS catalog, you will find about 2,000 significant SNPs related to type 2 diabetes. So what Elisa did is she took all these SNPs, connected them to genes, then connected the genes to the pathways, and did some further processing steps, removing redundant information and annotating and connecting them, where a lot of the useful annotation actually comes from these same resources that Sarah just mentioned in her talk, or her part of the talk. Can I have the next one? And that's actually the last one. And then it becomes something like this. Now you will probably, your first response will probably be, oh yeah, this is one of these hairball pictures that network biologists tend to show me and have no clue what I'm looking at. Normally I would start to use this as the start and then focus on what is there really in there, but actually there's a lot in here already. Like the part that is in the blue rectangle here is variants, right? The green are variants that occur in just one gene, one pathway on the lower section, and then there are a few variants that occur, two different variants that occur in two different genes in the same pathway, but still not really interesting yet, right? But the thing is, in that whole blue rectangle, there isn't that much, right? Most of these variants actually occur in the larger network, where you see now I have genes that connect to pathway, the SNPs that connect to genes that are in a pathway. But here, for instance, you see one gene where we actually have 6 different significant SNPs found in GWAS for that same gene. And you have one pathway that actually contains a lot of genes that all have, well, most of them actually have multiple. I'm not sure they all have multiple, no, not really. Known variant that contributes to the disease. And then if you go to the heart of this network, you see there's actually a lot of genes that not only have a lot of SNPs, they also have a lot of connections to pathways. So they occur kind of at the heart of the known biology, and they occur at the heart of the regulation of the disease. And of course, now focusing back on these pathways, which I will not do here, brings you at the heart of the disease. So actually diving into the heart of a network like this, you can go somewhere. There's something else here that really is a lesson we learned from other types of omics analysis we did on pathway level. Like the typical thing with even transcriptomics data that is even stronger in this type of GWAS related information is, how significant does something really have to be considered real, right? There's always discussions about GWAS studies like that. And people tend to tell me like, yeah, basically, if you really want to be sure, you should basically measure everyone, right? You need at least 100,000 participants in a GWAS study or more to really be sure. That is because you're looking at an isolated variant related to a very complex phenotype that will typically have minor effects anyway, and that is then also influenced by all the other variants that occur in the genome, and of course all the environmental factors that occur too, which makes it really hard to find significant results and then assess a meaning to it. But doing it on this level, you get to, you know, wait a sec, if I see this many significant genes in a specific pathway, and actually all these other variants that were found in this pathway and that by themselves were not significant, actually might still be relevant to look at, right? Because now I have a reason to filter down the complete genome space to a limited number of really relevant processes. That is something we typically do in transcriptomics. That is why we often say in transcriptomics, we can skip the typical false discovery rate corrections if we do pathway analysis, because the pathway enrichment itself is a kind of next statistical step. And you could start doing that for variants too. And I think this is my last slide.
Yes, thanks. So to summarize, there are rich sets and varied tools available for the prioritization of variants in rare disease studies. Standards, as I keep saying, are essential for results comparison and data sharing. VEP provides a convenient way to annotate and prioritize your variants with simple access to aggregated public variant data and phenotype data. It's quite extensible. We've talked about the EJP Rare Disease Project, which brings together tool and data providers to improve interoperability and simplify rare disease and variant analysis. And just 3 of the resources in here that we've talked about are Ensemble VEP, NextProt, and Wikipathways. So with that, I'd like to thank are collaborators with EJP-RD. So in particular, Freddie for providing slides, and Liddy Lane, who would have liked to have been here but couldn't make it, so provided some slides. I'd like to thank the ensemble team. And thank you everyone for listening.
Thank you, Sarah, and I'll check whether I can now see the questions again. The main question is actually, the only question I see, to be honest, and I know there have been more, is about the Nextprot Sparkle endpoint and how many users that has, and whether the Nextprot has been integrated in Wikidata to use the Sparkle queries.
So I'm afraid I don't have usage stats on Nextprot. That's Liddy's and she isn't with us. So we have included it in Ensemble VEP using a plugin which contacts the Sparkle endpoint. Obviously this does one query per variant, so you wouldn't want to stick a genome through it, but once you have a small set that you're interested in, it's quite useful then.
Yeah, I can answer. Well, I don't have the stats either, but I can answer a little bit more. First of all, Lydia has done wonderful work, not just on the RDF that they have there and the Sparkle endpoint, but also provides a lot of example, both direct and federated Sparkle queries to that resource. The federated queries basically already allowing you to do approaches that you would typically use a combined resource like Wikidata for, which was part of the next question. I think Nextprot is actually in Wikidata already. I'm almost sure there. That Wikidata part of the question relates to something we also did in the EGP, like how do these federated queries actually work? The typical thing, if you really work with RDF from multiple resources and you start to create Sparkle queries for these, is these Sparkle queries first of all become really complicated, and then you often run into problems because the different resources, even when they use RDF and correct URIs, still use different identifiers for the same thing. And to expand from one part of the query to the next, you need to somehow translate that. And it's hard to do that on the fly. So we're seriously looking into federated approaches that actually become more modular, where you query individual resources in which can actually then still be queried from Wikidata in relatively simple API. No, sorry, in relatively simple Sparkle queries that you could put behind an API, and then combine them all into a workflow that you could do in Jupyter notebooks, in KNIME and so on, and make these connective steps that you need to get from one type of ID to the other, or one ontology term to the other that is related to it, actually explicit in such a workflow. And I think that's really important work if we want that whole, because that's of course behind this question too, right? We have this RDF and Sparkle and open link data based approaches that are so nice in principle, and we still don't see them used that much. So what can we do to really move that work forward? The unfortunate thing is that it really is the only question I see. Does anybody else see more questions?
There were the answered questions tab.
That's a good idea. And then?
So there were two questions. One was related to the Perl API and if there are any plans to move to a Python API. The other one was if web supports breakend notation.
Thank you, Anya, for responding to those quickly. Great, we finished on time.
I think so. I think we.
So unfortunately, there are also no other questions on Slack. But thank you very much, Sarah and Chris, for enriching the environment with your talk in part 2.
Thank you.
So we will have another 5 minutes break now, let's say 6, start again at past. Unfortunately, we have been unsuccessful so far in finding the next speaker. So if there is no success story coming in the next 6 minutes, then we decided spontaneously that I will present about the Swiss Personalized Health Network project that I am running, which is the Swiss Variant Interpretation Platform. That is a clinically driven platform to annotate and interpret variants in oncology. So I hope we can find our speaker for the third part, but otherwise I will jump in for this.
Okay, it's 3:30. So welcome back after the break. So as the next speaker, we have the workshop co-organizer Daniel Stekhoven. So to give you a bit on his biography. So Daniel studied mathematics, so he's then a real theoretician, and then at the ETH Zurich, and did also his PhD at the ETH Zurich. After his PhD, he started shortly a company to do consulting in statistics before he then became the head of the Nexus, which is the central unit for clinical bioinformatics at ETH Zurich. And he's now presenting on his on the Nexus means of variant interpretation. So Daniel, the stage is yours.
Thank you, Sven. I hope you can hear me well and see my slides. So this presentation will be structured in two parts. So I will start off with a bit of background information on why we set out to set up this platform for oncology. And then in the second part, I'm actually showcasing the system. I know that you should never demo anything live, but we'll just try to do it. Okay, so I assume that many of you know this picture with the poor quantitative scientist sitting somewhere at the lower end of a funnel where all the big data is poured in since we have had the advent of next generation sequencing. So we sequence a lot of stuff. We're processing the data, aligning them. We're filtering, we're reviewing, we're validating. In the end, we try to annotate and predict. And at the end, somebody needs to interpret and report and produce a report. And this report has to be super sleek and super thin. And it just has to be, of course, always right. Because in the end, it might end up in a tumor board. And this tumor board reserves 5 to 6 minutes, maybe 10 minutes per patient to decide what treatment strategy might be the right one to do. So what's the big deal? Well, there is like gazillions of ways to arrive at an interpretation, something to support your final decision, what you want to put into that report. But it's not so easy to decide where to look, isn't it? So essentially, when it comes to clinical application, we are looking mainly in this finding for three things. First of all, it of course has to be super fast, because if we're speaking about a metastasized melanoma or so, we are just have a couple of days or better part of 2 weeks to react. It of course needs to deliver insight. If it's just a nice association that has no clinical relevance, then we can save putting it into the report. And it needs to be accurate. So it needs to be on target in what we're saying in these reports. So what could we essentially improve about variant interpretation in a clinical setting? So this project, as opposed to many research projects, has a very strong diagnostic side to it. So I am also group leader in the Swiss Institute of Bioinformatics, and we set out a couple of years ago, and by some magical capability, we were able to bring many different hospitals from Switzerland together into one room. And we fed them lots of coffee and sandwiches and asked them, so what do you need? What do you need to make variant interpretation better? And they came up with a wish list. For instance, it would be so much better if we just have one place to look at. It would save us time. Everybody wants to post the variants they find and add the annotations they themselves infer from what they see in their patients, but nobody has time, you know? So everybody wants to help, wants to post back to ClinVar or post back to to Civic, but they don't have time. So it would be great if you could help them with posting variants. Of course, the clinical bunch, they have their own art, you know, and what they say is that if I have a colleague, I'm an oncologist from Zurich, and I have a colleague in Basel, and he actually believes that this variant should be interpreted in this disease context in that way. I trust him more than if that is done somewhere around the globe. This might be some kind of Swiss chauvinism, but I believe that this is also true for other national territories and the clinicians inside. I cannot back that up with paperwork, you know. But anyway, they also want to have mutually approved annotation. And here is the interesting thing. When we set out with this group and we started working on convincing them what they need in this tool, you know, they all said, no, but why would I need improvement? I'm already annotating variants. Excellent. I have my tool. I have my favorite database. So what we did, we made a round robin, a very small round robin. We sent them a couple of sets. I think it was like 6 or 7 sets of variants for a certain type of disease. They didn't even have to process any data or so. They actually just got the variants and the disease. And we asked them, can you report on this? And we ended up getting total entropy. So we actually had variants that were assigned in one hospital, they were assigned pathogenic, and in another hospital, they were assigned benign. And in the end, we found out it's actually a sequencing artifact by the Eintorrent sequencers. This kind of made them more susceptible to our ideas to set up this variant interpretation platform. To set up the database, we are actually at the moment working on a ramp-up data set. So our partnering institutions provided us with variant data and their corresponding annotation to the variants. And at the moment, we are managing the discrepancies between, as I just said, there are discrepancies between hospitals, and we're managing those. And ultimately, annotations of variants is a moving field, a moving target, I mean. So if a variant that has been of unknown significance or has been tier 3, if we're speaking about clinical significance, can now have a different annotation and also a new application, especially if we speak about drug-centric interpretation of variants. And they want to basically be notified that if they previously have submitted from one of their patients a VCF to the database, that they get updated. Oh, by the way, this annotation has changed. Maybe you can moderate your treatment decision based on this information. Well, and practically speaking, taking all these wishes together, what we need is we need some way to submit variants to this tool. We need some way to curate variants inside this tool. And ultimately, we need some way to retrieve the variants from the database, the annotations, I mean, from the database. So these are our front ends. And on the other hand, we also need the place to store the variants, a place to store aggregated evidence. Aggregated evidence basically means annotations and interpretations from other resources, like other repositories. And what that means, I will show you in a minute. And our own interpretations, which then, which is basically also evidence, but we differentiate it since it is from our platform, which we then hope we can share with back to the other places as well. So these are the backends. In between, we have an API that serves the front end. And this is our system. Of course, there is many more aspects to it, but I will save you with these details right now. The variants are coming mostly from the large university hospitals and smaller institutions. The aggregated evidence we have collected, being inspired by the G2P aggregator. Our harvesters are now a bit different, but will still help us a lot in the beginning to get an idea on how to collect the data. The interpretation will be done by oncologists and hemato-oncologists in Switzerland. So we have identified an expert panel, and they will get curated annotations, and then they can basically, like in a Tinder app, they can swipe left and right whether they agree or disagree with an annotation. And the moment they swipe left, we actually ask them, so why? On the front end side, so again, submissions will, in a production version, will be fed also through a API, hopefully. The curation part is taken over by the Swiss Institute of Bioinformatics. We were able to recruit several very skilled curators, which are also associated with SwissProt. We heard that one before. And the retrieval front end has two flavors. So there's going to be a clinical front end and a research front end. The research front end essentially is just a public front end where we will put just the interpretations in. But the clinical front end allows you access to the actual clinical data. And with the current laws in Switzerland, it is not possible that the access to this data goes beyond the medical secret. So this layer is specifically only available to oncologists and pathologists who work in a hospital and have a authorized and authenticated identity with said institutions. And this is how we actually were able. I mean, this is maybe also an important piece of information for some of the cores out there. If you want to make the sharing and the access and the communication between different hospitals possible, it might not be possible if you have kind of like a network or a communication tool that is not open in the clinical sense, how MDs see it. But if you put it under a medical secret, suddenly everybody is totally happy with it. I'm not saying it's an easy task, but that was the door opener for us, and we're working on making that happen right now. Okay, so this is just when I share the slide right afterwards. So that was the first part, briefly pitching about the history. And now I would like to show you what it looks like. So let's pull open Chrome. Good. Okay, so if you go to swip.ch, you see the database in full swing. Here we have no data in it because we are still working on our first batch of our own annotated data, but it already gives you access to the gathered data from the public resources. There is also information up here about different releases, what were the changes. We have a statistics page, which shows you from which resources we have pulled which variants. And currently, as you can see, we have, are you seeing this? Can you see the website?
Yeah, looks fine.
Okay. Currently,
Yes.
we have 4 resources that we are querying. We are in the process of extending this, including other resources that offer us API access. And ultimately, here you can read up on the history a bit more. Of course, here again, the wish list, our partnering institutions, and the main PIs on the project. Okay, now, of course, I want to show you a bit more detail about the functionality. So let's go to the test server. Here, you will see it's less genes and less variants, but at least these are genes and variants of which we were able to find in the data that was provided to us from the hospitals. So as you can see, I logged in with my own account. I have increased permissions. So I can, for instance, start querying a gene here. So let's query a gene like CSF3R, and then I get selections. You see different potentials. I can go to the gene page. For this gene, we have only one variant. If it's another, you can try BRAF or so, then you have more variants. But then I can basically go here to show details, and that's what I end up having. So let me walk you through this view here. Up here, we have basically the location, the standard information about the gene. You can also look at several aliases. We are right now integrating other location aliases beyond HGVS. We are also integrating, as we speak, the variant representation hashing for variants, which will also greatly increase our interoperability with other resources. Then let's skip the SWIP information for 1 second and go down here to the public available information. Here you can see that obviously Civic has a piece of evidence for this gene. So we see that the drug ruxolitinib obviously has at least a C or D evidence level. We summarize all the findings for each database in these evidence level graphs. That's kind of also where we deviated from the original G2P aggregator. So the G2P aggregator tried to have a harmonized score for the different resources. And we decided not to go down that road, rather to show as is what the different databases provide. Again, the pathologists and the oncologists, they kind of have their favorite database, which they want to have as a reference, and they are familiar with the coding or let's say the ranking of these resources. You can also extend the evidences to have all the references listed in the right order. And if you hover over an evidence piece, you will actually get the title, the short list of authors, the magazine, the journal, sorry, not magazine, and the start of the abstract. And as you can see, we have a text miner that goes through these evidence pieces and identifies gene names, variant names, and disease names based on the text mining that we do on the information that we feed to it. So we always use variant, gene, and disease. Down here, we also have additional information for said variant. So some algorithmic impact prediction, polymorphisms, and this is kind of like poor that we don't have the SPO here. So let's briefly query another variant. Let's use our best friend here. So when we query this, you see down here, that's another project from the Swiss Personalized Health Network, where they offer protein structures. So that's the wild type protein structure of BRAF. And it shows me the location of the 600th location of the amino acid V. And based on this, let's say a very skilled oncologist or pharmacogeneticist or so can maybe make a statement about the usefulness of certain drugs. At least that's what they told me. If we have something like BRAF V600E, you see there's much more evidence pieces here. And suddenly these summary informations that we display here are more useful to get an idea. Of course, also here you have the evidence pieces, first of all, summarized. I can select a certain disease to filter first. I can only select one disease at the moment. And I can also collapse these to get all the information. So since we do not have any curation on BRAF, I will briefly go back to our previous variant. That's why I chose it in the beginning. So let's come to the SWIP information here. Now the SWIP information that is separated from the public one because we call it our own annotations. And you can see that there's a little bit going on here. So for instance, for chronic myeloid, for this one here, we can see that there was some curation activity going on. We always have a logging. So we see here Anne Österreicher is the curator from Swissprot. She actually did this beginning of this year and selected a pathogenicity, which is unreviewed, and also a clinical significance level. So let's go and look at this. When I open this up and I see the evidence pieces here. No, that's another one. I see the evidence pieces. They have not yet been submitted. The reason being that they are all in draft version. So in the public interface, you would not see them yet until they have been reviewed. Now, since I am in God mode logged in here, I can actually go and look at the curations that she did. And we can see there's a lot going on here. So she actually found different publications over here. So one for this gene, which is obviously a diagnostic type of evidence. We have one here, a predictive evidence for dasatinib, and so on and so forth. I can increase the information content on this and look at whether there were some comments from her side on what she found in there. So they have the most of the inputs, and I will show you this in a second, that is structured, but they have also the option to add comments for context. And down here they have comments for their colleagues, which there is none here. I can see them only because I have also curator privileges. Okay, let's look at what happens if we start to curate something. So if I click here on curate, I get taken directly to the gene name and variant, location, and then we have here pathogenicity and clinical significance. Probably these two will disappear. In an early phase, we had disease up here as well, realizing then that actually the diseases should be put down here because a variant means something different in a different disease. So if you see here, these are the evidence pieces that she added. If I go down here, I can see that I have now the option to either add another evidence piece. So maybe you Googled or used some other means, or I just read a paper and realized, oh, this is a fantastic piece of evidence for this CSF3R gene. So I can add the PMID here. Additionally, we have our text mining tool. So if I click here on the text mining tool, what I will get is a list of publications that have been selected based on the gene, the variant, and if I did select the disease. So down here you see now I can expand the score, and we see the text mining faucets that were selected. So we have several diseases showing up. The disease names are from the HBO ontology selected. Drugs, we have not found drugs for this publication, but we found the gene. We found an alias for that gene. We obviously found some age groups and gender groups. Based on this, the paper gets associated with a certain score, and that is a first go at sorting these publications. So I can now say, oh, yes, that looks interesting. Let's add this publication to the list. And here I end up now, I have now selected the evidence piece I want to annotate. And now here comes the curation part. I can decide now which disease it is. So let's say we had this myeloid, acute myeloid leukemia. So let's say it's this. Whatever I'm filling in here is now totally bogus. So don't hold me accountable for this. If I go down here to the effect of the variant, there is nothing to select here. Also not down here, because based on what I select here, I get a different set of possible effects. So if I select a predictive type of evidence, actually I can select a drug, and that was by accident. Let's select a different drug. I can select a drug, and now I have here the traditional values for the effect of the variant based on it being a predictive piece of evidence. So I can change here to a prognostic piece of evidence, and now I will get the prognostic wording. So all these is structured information that the curators feed into this end. So let's select something. I then have for each of these types of evidence a different set of tier selection. So in this case, it's functional criteria. I can select something based on what the publication was about, and then I have, in this case, let's say it's somatic, and the support is a mean of the curator to say how much. I mean, we don't want them to use too much free text. Ideally, not at all. So we have to give them some way how much they trust in it. Again, here I can actually add complementary information for context. Everybody see, I can ask for comments by my colleagues. And ultimately, there is also a nice feature. So we can say that we found this one sentence up here that kind of supports absolutely my claim. So I can select this sentence and I can say, add this as a textual evidence bit. So we cannot see it, but now it is down here added as a sentence, and it can be queried through the API afterwards. This is how we do the whole curation part. I can then save it as a draft to either have it reviewed by my colleagues whenever I change something. So now it was just created. So let's say I just realized that I fumbled the effect. Let's say I update the draft now, and in the history I can actually see who changed what, when, to what end. And this also is if I now save the evidence, I will see it was saved from a draft to a saved evidence piece. And when I go back now to the variant overview, you just see that AML just now got this bogus entry by me, which we have to revert after the talk. One of the aspects that we had to, or that we want to serve here as well, is to not only improve, but also to offer a helping hand towards the hospitals. So give them an opportunity that when they encounter a variant in a patient that proves to be very difficult to interpret from their side, that they can actually request curation from our data, but from the curators in SWIP. So to that end, we have built a dashboard, and we're still working on this loading here. It's loading because it's loading way too many things right now. But essentially, a clinician can request a variant by us to be curated. They can assign an urgency to it, so a deadline. And no, it won't always be the urgency is super urgent, because in the situation of, for instance, heme cancers, there's usually more time to react on a certain thing. But in the end, our curator said they will be able to respond to high urgency requests within 2 days, working days, that is, at the moment. And down here, basically, you see there the work in progress. So these are all the drafts which are in the system. And as you can see, they are mostly for one gene, because usually you end up reading a publication on a gene that was investigated in one disease, and you end up having different, you have one gene, you work through one gene that we found, and you have different variants, and you end up adding all these different publications stack-wise. Now, this is one way to do it. This is kind of, at the moment, the staple works of the curators. They just need to fill up SWIP with annotations and informations. Eventually, our expert panel needs to review all these annotations. Once they are reviewed, we will release them to the world, except they get put under some kind of publication lock, which we had to grant to some of the clinicians, especially in the heme oncology, because there are no real well set up resources for hematological cancers at the moment. While up here, this will be kind of like the very specific one variant, one gene requests coming from the hospital that kind of usually will have a higher urgency. Now, we are on GitHub, and you will note we've discussed how many private labels we have here. We're still working on improving that. We hope that no later than the end of Q3 that we can go public with the code. There's still some vanity from the software developers that everything looks nice. I ideally hope that we get it out as fast as possible. Furthermore, we have a REST API at the moment, but it's basically the API that is feeding into the front end. So this API is under heavy development. So we just now received several requests from other SBHN projects that they would actually like to pull our annotations, which we queried from public instances, because we already did it at one place in Switzerland. They actually want to pull the annotations from the other places through SWIP. So we will rather sooner than later have a public API that will be more stable. It's not feeding into the front end, but will be queryable directly. Of course, also supporting variant representation. And while we're already speaking about GA4GH, we also have a beacon on top of SWIP. But since there is no SWIP data yet released, there is just very humble feedback from it. And so if you're interested, here is the text miner. So this is the tool that was developed by Patrick Ruch's group at HESO. And you can play around with it. Just bear in mind that the Swiss Variance Interpretation Platform is actually querying this all the time in the back. As soon as we have something, we're building the faucets. So it can take quite some time to kick off the text miner. So let me briefly switch back to the presentation here. So I will share these slides later on the Slack channel. You will see, so SWIP is available under swip.ch. I promise that we will go public soon. We're also actively contributing to the Beacon version 2. Which will make our dear harvesters probably obsolete, but let's see where that goes to. I showed you variomes, and there is also a YouTube video about SWIP, which you don't need to watch anymore because you just saw me present here. And a shout out to my colleagues. So Faisal Al-Qudami and Vipin Sridharan are my software developers and data managers who have dearly set up this system. At HESO, we have Patrick Ruch, with Emily, Anais, and Deborah, who are working on the text mining aspects of SWIP, and the whole backend, as well as the curation and the connection with the clinical side, is coordinated by Valerie. with Florent, Dylan, Anne, and Livia. And of course, I thank also the guys who initially made this so efficiently possible, which was the SIP working group. You remember the guys that we fed coffee and sandwiches to for somatic mutation calling. And I'm happy to take your questions.
Thank you, Daniel. I think there are 2 questions in the portal. Let's start. Well, they're both from Farid Musa. First question, will this platform be open to the world or at least to the hospitals and clinicians somehow accredited to contribute to the curations, or is it limited to Switzerland and or Europe?
So in a first phase, I mean that there's two aspects to this question. We will publish all our own interpretations or annotations, similarly to what Civic does, similar to what OncoKB does. We will publish these to everybody. So this will be swip.ch. You can access the website right now. So we will publish these. To contribute on the curation side, this ruins our strategy to have also a communication tools for this validated for this trusted validation thing. So what I forgot to show you actually. We'll still have some time. So let me briefly go to the development version and show you that as well. So 1 second. So what they also wanted to do is they wanted to actually be able to look up whether a fellow colleague had a similar case. So basically this variant, and maybe all the annotations in the system are maybe unknown significance, cannot tell, moderate support, whatever. But they see, oh, by the way, there is actually my colleague in Geneva, he actually saw the same variant, you know. And so when they now say, okay, so I have this variant, and what they have is this ribbon here. So they can actually go to samples, and what they will see here is they will see information about the samples that have been submitted to SWIP so far. So they will see some information about the patient, the institution, and they can directly contact their colleague and ask him, if you click here on the contact, the information about the patient will be added to the communication device, which is not going to be email as insinuated here, but we'll tell them, hey, by the way, your colleague is asking about this patient that you had previously. They had the same variant like I am observing now in my current patient. So I would like to know how you treated this person, because there is obviously no tangible evidence available. And as I told you earlier, the only way we are able to do this is through the medical secret. So this ends at the national border of Switzerland, this medical secret hotfix. And we also do not intend to replace resources like Civic that have a crowd curation philosophy associated to them. It's also that, as I alluded earlier, we are not a pure research platform, but our first priority is improving diagnostics in Switzerland. So this is, sorry, a very long answer to the question, but it's a tricky thing. But we are also sharing our code so other places can improve, other countries can set up similar systems in that essence. And we take great deal at being as interoperable as possible. this is why we adopt the GA4, GH standards. This is why we try to build as many on the shoulders of existing ontologies, that one time when we start sharing all this information across the whole globe, which I hope will be rather sooner than later, then we're ready.
Okay, thank you for that very long and quite very reasonable answer. There's an additional question from Farid Musa asking, for some reason I see 705 genes but 0 SWIP variants.
Yeah, so this is somewhat buggy. So the 705 genes are all the genes that we no, wait a second, you mean SWIP.ch. So there is no SWIP data in it, and the 705 genes, that is a accumulation of, I believe, OncoKB and Civic. These are all the genes and the 300,000 watt variants that we were able to harvest from OncoKB and from Civic in a proper quality. But there is no SWIP variants in it because we have not yet released any. In the SWIP test version that I showed you, there are less genes and less variants because we based the select. So we took these 705 genes and 300,000 variants, and we only picked the ones that we already saw in the submitted ramp-up data we had from the hospitals that have contributed thus far.
Okay, and the last question by Julia Pereira. Thank you for the presentation. This is a great effort. Are you planning a patient-centered query instead of gene or variant to ease the process for the clinician to decide on a treatment for a patient?
Absolutely. This is ultimately the goal, which will facilitate the job of the pathologist or the oncologist at the clinic. So what we will do, we will build an API towards the hospitals, the partnering hospitals. So first of all, they can upload the VCF, and what they will get, they will immediately get a snapshot of the database consisting of the annotations and interpretations from said VCF. At the moment, we're not allowed to use mutational context in the interpretation. We're only allowed to look at one at standalone variants, but the reporting we're allowed to do. So we can report on the single variants in a VCF, and we will do that in one report. Of course, this report is not a PDF or anything. It will be a JSON or XML excerpt from the database that goes directly to the hospital. So this is then going to be the patient-centric report, which they can also look up directly in the system and have it be connected with the resources that we queried.
Okay, thank you, Daniel. There are no more questions now in the chat.
Oh, there is one by Chris. Let me ask that one because it's important. So yes, we totally want to link up with others, and I also we totally want to make these tools available as soon as possible for others to be used, and we are more than happy to basically collaborate on improving the tools or discussing about the best way to have the API set up to talk to you guys. So just shoot, come to me and we come to you and everything. I all in on collaborating on this.
Okay. Now there are finally really no more questions, and we come to the end of the workshop. In theory, the channel is still open for 15 minutes. So if you feel like interacting, please go ahead. But I think I will now officially close the workshop. I hope my co-organizers agree with that. So we thank you all very much for the participation in that workshop. And thank you very, especially great thanks to the speakers for sharing their very interesting work. And those of you who stay at ECCB, enjoy the virtual conference and see you at the next occasion for the annual core community workshop in bioinformatics. Have a good time. Bye-bye.
Bye. Thanks.
Thanks, Mitch. Have a great day.
Take care.
Thanks, Sarah, Chris, Camille. Thank you all.
Thank you, everyone. Thanks for stepping in, Daniel. That was great.
Thank you so much. Bye-bye.
Yeah, thank you for the organization. See you around.
I will post the invite link to Slack again in the forum. So if you want to join us on Slack and continue discussing and also reach the speakers at a later time point, we're really happy to see you and welcome you in the instance. Have a nice day. Bye.